Okay, good morning, everyone. So we're going to start. So thanks a lot for attending. So as usual, we are on a webinar mode. So those who attend online, you can report any technical issue in the chat and ask your question, please, in the Q&A tab rather than in the chat. And we will allow you to speak during the discussion section and interact with the room. And with that, I thank Isabelle for introducing today's speaker.
Thank you, Jeanne. So it's my pleasure today to introduce Elizabeth Wolkovitch. Lizzie is an associate professor at the University of British Columbia in Vancouver. She's professor in forest and conservation sciences. Previously, she was assistant professor at Harvard University, where I met her for the first time. And before that, she did several postdocs at the University of California, in Santa Barbara and San Diego. Lizzie has very broad interests: community ecology, ecosystem ecology, invasion biology, evolutionary biology, and phenology, climatology, and global change. And today she will talk about the importance of time in ecology, in biological processes, and how global change is affecting the timing of those processes. Lizzie is currently visiting my group at CEF up to early April. If you'd like to meet her, please contact either directly Lizzie or me if you want to set an appointment. Lizzie, the floor is yours.
Thanks. Okay, first of all, many thanks to Isabelle for hosting me. It's been great. I was here a little bit in January. It's great to be here. It's been wonderful. Thanks to all of you for coming, and many thanks for all the excellent technical work. I assume you'll wave your hands wildly if there is a problem. And for those in the back, if at some point you can't hear me, or I start talking way too fast, which sometimes I do, feel free to wave your hand wildly and we can try to fix it. Okay, but today I want to get started by going way back in my career. This photo comes from almost now 20 years ago, in the first year of my PhD. That is me. And I'm in the habitat that I did my PhD fieldwork in, which is a habitat called coastal sage scrub, which is prevalent throughout southern coastal California. It's endemic to that area. And because it is prevalent throughout only southern coastal California, this is what much of the habitat looks like today. So this is an aerial view of San Diego, and hopefully you can see the habitat patches that are left in it. These little fingers and tendrils are canyons with some of the remaining coastal sage scrub that is left. These are little islands, if you will, within a massive urban matrix. And for my PhD, this is what I was interested in. I was interested in understanding fragmentation. And this has been a system that many people had worked on, this massive reshaping of space that had started, I would argue, mostly in the 20th century by humans, had led to the field of spatial ecology in the 1970s and 1980s, and really gave rise to a growing interest in understanding how the reshaping of space, habitat space, by humans could reshape biological processes. Specifically in my PhD, I was interested in understanding how habitat fragmentation had affected the invasion of multiple grasses. These are grasses, you may know, they are Mediterranean annual grasses that are major invaders throughout this habitat in coastal California. But what I came to find after 4 years of fieldwork was that it wasn't so much how the alteration of space, these sort of islands and mainlands that we had created and small corridors in between, were reshaping invasions as much as the invasions were functionally reshaping time in the system itself. And you can see a little bit of that here. These grasses in the front are right about to set seed or start going into from flowering to seeding, whereas the native species in the back, which are generally shrubs and other perennial species, have just leafed out a couple weeks before. So the grasses get a big head start. And you see this throughout the season. This is later, about 2 to 2.5 months later. And what you see here is that the invasive grasses have already started senescing well ahead of the native flora. This is Artemisia californica, which you can hopefully see in the back here, one of the major native shrubs throughout the system, which has just started to set seed. So the grasses nest several weeks at least ahead of the native vegetation. And so what I was really finding was that these invasions actually seemed to change the rhythm of the seasons, how the biological seasons were progressing. And I would argue that humans aren't just changing space in really major ways, but that we have fundamentally altered biological time. So this invasion case is one special case, but certainly on a global scale, I would argue we have massively changed biological time through anthropogenic climate change. So this is just a quick plot of cruise data for the Northern Hemisphere temperature anomaly going back to the 1850s with warming or temperatures on the vertical axis. And the biggest impact, or I wouldn't say the biggest, the most reported impact that we see of climate change is the alteration of biological time. And we see that in records such as these. These are wine grape harvest records from France, which Isabel has previously worked on, and I have as well. And so what you hopefully can see here is these are the records going back to the 1600s. They go back further, but these are what we considered more high quality. They are relativized at 0 to September 1st. And all I want you to take away here is one, there's immense variability in these records and the timing, but also at the last 40 years, you see this big shift downward towards earlier harvests of about 2 to 3 weeks. And so my lab focuses a lot on phenology, and that's what I'll talk about today. Most of you may know this because I'm in Europe, but in North America, I always have to remind people that phenology is the timing of recurring life history events, such as the day a bird arrives at its nesting ground, or the day of leaf out or flowering for plants, which is what I predominantly work on. And I want to make a quick case that understanding phenology is one of the most critical things that we need to do to understand and predict populations, communities, and ecosystems at both a fundamental ecological level, but also to better mitigate and adapt to climate change. And I'll go through a couple quick reasons for that here. The first one, starting in the upper left, is a field site I worked at in Quebec called Saint-Hippolyte in the Laurentian Mountains, but it represents a really big source in the boreal forests of how forests store carbon. And most of the land surface models that we use today assume that as growing seasons lengthen with warming, these forests will store more carbon, and that will actually mitigate climate change itself. So how well we predict phenology also impacts how well we predict climate change. The next one, I don't work sadly on the porcupine caribou herd, but this is an example of some of the work I have done on trophic mismatch, because one thing we're seeing is that as you look at species or populations that are better able to shift their phenology with warming, we also often see that they perform better, grow better, or even have higher fitness with warming. And the reverse is also true. So there appears to be a potential to predict the winners and losers of climate change, if you will, using phenology. And then finally, as I'll talk briefly about at the end, I think the more we see extreme events, the more we understand that to predict the impact of them, we have to know at which biological stage they're at. So these frost events are happening partly because so many grapevines in France are at a very early leaf out stage at the same time. Those leaves are more vulnerable to frost during early leaf out. And these heat damage events are having the biggest impact on growers when they hit during flowering, because generally we see the grapes aborting their flowering and their fruits at that point for their flowers. Okay, so what I want to talk about today through this lens of phenology is 2 stories about how I would argue that climate change has first stretched out time and what that means for community assembly. And then I'll dive in on predicting phenology of woody species and talk about how it has accelerated time. And very quickly, if we have time at the end, I'll tell you a little bit about where my research program is going next in these areas. Okay. So to get started with stretch time, I want to go back to where I did my PhD. So again, this is just a photo of aerial San Diego with little islands of habitat, these canyons that are too steep to build on, buried within a whole bunch of freeways and housing. And when I started trying to think about what does it mean if climate change is changing time, what would we predict to happen to populations, communities, and ecosystems, what I realized was that we have a lot of theory to draw on to predict what happens when humans alter space. So I, as a PhD student, got to use lots of island biogeography, metapopulation, metacommunity. We have a whole theory of how edges should work, as well as the design and type of corridors. And when you start thinking, okay, what theory will I draw on to think about how humans are altering time, I would argue you're left with many fewer theories to look at. But there is a major one, and it's a major one in community ecology that I'll run through quickly here, which is the storage effect model. Now, this is a model that uses interannual variability, generally in the environment, to promote coexistence. And very quickly, I'll run through how it works with a very fun cartoon, not fun, but very simple cartoon, where I have 3 years and relative abundance on the vertical axis. So the storage effect model basically says that multiple species can coexist in one environment as long as they have slightly different responses to the environment, and that those responses co-vary with the amount of interspecific competition they experience. So in this case, species A does really well in year 1. This is a high population year for it. It's very abundant, it has high interspecific competition, and it persists at a lower level for the next 2 years. The model says that it can coexist with species B, which has a slightly different response to the species, so that year 2 is better for it, and that it persists through the other 2 years. And so on and so forth. You can pack species into communities through this model, as long as they have only slightly different responses to the environment. And this was a revolution back in the 80s and 90s when people started to understand it, because it meant that species could be extremely similar in how they might look on a landscape. But they could just be responding in small ways to the environment differently year to year, and thus they could all coexist at once. And so this model has generally been applied to time on an interannual scale, but there isn't any reason, and Chesson himself says this in some of his early papers, that it wouldn't work within a growing season, so on an intra-annual scale. And so I set out to look early in my postdoc period for evidence that you see species partitioning the growing season, and that you see evidence that perhaps this intra-annual scale matters to community assembly. And for that, I worked on invasion biology. It's what I'd already been doing. And I would argue it's also such a fundamental set of questions in community ecology. It helps us try to answer, what are the characteristics of a species that allow it to invade a community? And what are the characteristics of that community that allow it to be invaded? Which is also fundamentally what we do when we try to understand and predict from storage effect models and like. And so I came up with several hypotheses, but the main one I ended up working on is this idea that was already in the literature that invaders should be more plastic or flexible in how they respond to many things. But if we think about what that would mean with climate change, and I have 2 hypothetical cartoonish niches of species here, the exotic is in teal and the native is in purple. What's happening, I would argue, with climate change is we're stretching out these growing seasons. We're extending the biologically available time for species. And if you expect that the exotic species is better at being flexible as the season extends, you would expect it to track that better. So it would get an early opportunity to use resources, effectively a priority effect over the other species, and might be a way that invaders are establishing and being successful in communities. And so I, many years ago, ended up testing this with a synthesis of long-term data. I went out and got many data sets, but I only found 5 where we actually had a good number of both native and exotic species within a community, and also phenological records over a substantial period of time. These are the 5 sites, and these are long-term data. So this means there's a little picture in the bottom to tell you that people had gone out and written down exactly the same phenological event for a number of species for many years. I think the minimum years we had here was 20 or 30. I'm going to show you the results just for the 2 sites that I've continued to work on off and on, and for speed. What we ended up looking at, sorry, this should say shift in days over time, a little over, is we ended up looking at what is the amount of change over time for the exotic versus the native community, given that we expect that the exotics might shift more if they are more plastic, and that's part of what's potentially helping them invade the systems. I showed 2 different versions of the model. First of all, I should say the native species are in blue, the exotic species are in red. And there's a version of the model that is just a basic simple regression, and we also phylogenetically corrected for different evolutionary histories between the exotic and native communities. And what you see in both, these are shift in days over time, is that in both cases, both for Concord, Massachusetts and Chinor, England, there's been a significant advance that is higher for the exotic species than for the native species. So we do see evidence that exotic species tend to be more phenologically flexible. We've done work in the lab since then that shows, I think fairly convincingly, that this is a trait that is present in the native range, and also that it does lead to a priority effect potentially that is driving potential differences in competitive abilities. Okay, but what I've been working on more recently is trying to understand if these sorts of patterns scale up and matter beyond invasions. And for that, we've been doing a more modeling focused approach. And I've been working with a colleague of mine called Megan Donahue. She is both a marine biologist and a storage effect modeler who works at the University of Hawaii. And we've been trying to adapt the storage effect model so that we can use it to try to understand if there would be more general expectations that as you shift phenology and as you have climate change in a system, you would expect these traits to matter. And so the way we've done this, oh, I should say before I forget, sorry. Oops. I'm not going to talk deeply about this model, but if anybody wants to chat more about it, I'm happy to. It is a storage effect model that includes both intra-annual and interannual time, and species coexist through differences in their relative germination rates over the period, as well as fitness differences. And what we did to try to alter how species are shifting their phenology possibly is we altered effectively the germination kernels within these models. So every species in this model has a germination curve that is unique to it. It looks like this, where the germination rate for each year, the G sub I, is on the vertical axis, and a metric of how time is working in this model is on the horizontal axis. And so every species has an intrinsic shape of the curve, but also importantly has this tau I parameter that controls, as you can see, when it will maximally germinate relative to this abiotic parameter tau P. So tau P in this model is the start of the season, the start of a resource pulse. And depending on how close tau I and tau P are, the species will germinate different amounts each year. And so in this case, tau P is slightly later than tau I. The species doesn't germinate at the maximum. But in another year, if you had them be the same, so tau I equals tau P, the species would germinate at its maximal amount. Effectively, what we did in the model is altered these parameters so that we had a phenological tracking model where tau I would track tau P to different amounts. And that allowed species to effectively match to the environment to different rates. So what we then looked at is how temporal variability would affect coexistence. There's some obvious outcomes before you even start to run simulations from this model. And one is that high trackers match the environment best. So species that alter their tau I closely to tau P obviously win. And given no other variation, they would always be competitively dominant and outcompete any other species in an environment. So you obviously have to add something. There has to be a trade-off. Otherwise, the world would be full of one species that phenologically tracks perfectly. And what we added was what we felt there was the best evidence for in the literature, which is a trade-off where species that track better are also those that are poorer resource competitors at low resource conditions. So effectively species that have a poorer R star value. So this is effectively almost a competition colonization trade-off, but applied to a different type of model. So species could vary both in their R star and they could phenological tracking parameter. And I'll show you the results now from model runs where we ran the model for 500 years in a stationary environment. So the annual resources varied year to year, but there was no trend over time. And I'm showing you this with the two traits that we allowed to vary. So the ratio of R star here on the vertical axis is species A over species B. And the ratio of tracking, how much it can adjust based on the start of the season, is shown on the horizontal axis. The green dots, and these are two species communities, the green dots are all of the different runs that we tried, and the blue dots are those where both species co-occurred by the end of, I think, a 500-year run. And what you can obviously see, which is not surprising, is that there is a trade-off here, where in one corner, the species groups that are persisting are where species A is the better tracker and species B is the better competitor, and the other corner is the reverse. This makes perfect sense. There's a trade-off where species can coexist such that one is better at tracking as long as the other is better as a superior resource competitor. But then we wanted to know specifically what would happen with climate change. So I just showed you the model runs for the first 500 years. We then adjusted the system so that the resource pulse started earlier and earlier for the last 500 years, a non-stationary environment. So this is tau P, the timing of the resource in the system, and I'm going to show you the results now for the second 500 years. We left the systems as they were. They all established for the first 500 years, and then we looked at what happened after 500 years of non-stationarity. So here I'm showing you the same axes. The blue dots are the species pairs that coexisted after the first 500 years, but did not persist to the end. And the purple dots are those species that persisted after 1,000 years, including this non-stationary period. And so what you see is perhaps not surprising. There's no immigration in this model. So what's effectively happened is that climate change is twisting the trade-off space but the communities have already filtered based on a certain trade-off. So we get fewer species pairs left at the end once they're filtered through this climate change environment. And that was interesting to us. It suggests that you might have fewer species pairs that can coexist fundamentally through this trade-off unless you have extreme amounts of immigration or other factors going on. The other thing that we didn't see coming was this part here, which is in the center here, where the ratio of tracking is equivalent between the two species and the competition, our stars are equivalent, are what we would call equalized communities. They coexist not because they have the trade-off, but because they are effectively identical species. You have two species that are almost identical to each other and are not fundamentally coexisting, other than the fact that they're just the same species traits. And that means that potentially with climate change, you're getting communities that are not coexisting because they're different, the two species are different with a trade-off, but because the two species are effectively identical, that you're sort of reducing the trait space you would expect. Okay, so that is talking about why I would argue that climate change is importantly stretching time for biological communities with impacts potentially on plant invasions. And I think potentially really changing the mechanisms, at least that we have for the next several hundred years of coexistence on the landscape. I want to talk now about how climate change is accelerating time. And so for me, this was an interesting question because we've done work in my lab that says that this sort of tracking matters. We have evidence from natural systems that something like phenological tracking may matter to climate change impacts, but we want to actually understand how could we predict which species are tracking well and which species are tracking poorly. And so for this, we've worked predominantly on woody species phenology, because I would argue it's where we have by far the best data and the best understanding of the underlying environmental cues that drive it, where we might be able to start to build models such as Isabel has of what to predict in the future for bud bursts with climate change. And so I'll walk quickly through what these three cues are, even though many of you may know them. This is a hypothetical imaginary time diagram again, but here time is at the end of the summer or early fall, and we're moving through the winter season into the spring as we go a long time. And so the cues that we expect here is that as species become dormant in the fall, they then accumulate a certain amount of winter cool, and we call this chilling. And that at some point physiologically, when they transition and have enough chilling from endodormancy to ecodormancy, they then would respond mostly to cues that we call forcing and also to day length cues. And this combination of cues, chilling, forcing, and photoperiod leads to bud burst in woody species. And this seemed good for us. It felt like if we could better understand these cues, we could predict which species are better trackers. So we might say things like oak, where we already knew that they tend to have weaker photoperiod and potentially weaker chilling cues. These we would say are going to be better trackers. And species like Fagus sylvatica or beech, which have a very strong photoperiod cue, so linked to day length, would be poorer trackers. So this seemed like a great area for us to go, because then we could start to predict the actual tracking and think about what the assembly of communities would be in the future. The other reason we were interested in it is a reason that lots of other people are interested in it, which is in this very simple diagram. If you want to predict what's going to happen with the phenological response in the future, if you're only using forcing, and if the species only have a forcing cue, then you have this simple linear relationship, hopefully, where in the current period, you get a line that extends into the future, and it's very easy to make forecasts of what the phenology will be. But if species have chilling and photoperiod cues, then at some point, these may become limiting or shift the amount of forcing needed, and we would have a non-linearity that we expect in the future, but can't easily predict from the current without including these other cues. And what you would expect if these other cues are operating is that after some point of enough anthropogenic global warming, this curve would start to turn, and the response, the change in days per degree of warming, might go down. And that is exactly what started coming out of the literature about 8 years ago. So this was one of the first big papers that said, yes, species seem to be slowing down how much they respond per degree of Celsius to warming. And that led to a big and exciting debate about what cues it might be. So it went back and forth between it's probably chilling, to know it should be photoperiod, to know it should be not photoperiod, it's more likely chilling. This was great. My lab was super excited about this. I was excited. I was like, we do meta-analyses. We can come up with like a coherent answer for what does the literature say about which one of these cues is driving declining sensitivity. And before I jump into how we did that, I want to just show you what the underlying data for these early papers that talked about declining sensitivity was using. These are data that you may know. These are from the PEP 725 database. I'm showing Betula pendula, silver birch only here, and I'm only showing 45 sites because I want to use absolutely identical types of data. So these are the same 45 sites in the on the top panel as on the bottom panel, but I've taken out 2 different decades, the 1950s, which were quite cool, and the 2000s, which were quite warm, spring temperature on the vertical axis and day of leaf out. Spring temperature on the horizontal axis, day of leaf out on the vertical. And what you can see is the declining sensitivity. The lines are effectively much steeper when the mean spring temperatures are cooler than they are as the mean spring temperatures have warmed. These lines have gotten much shallower. So this is declining sensitivity measured usually in change in days per degree of warming. And the actual difference across these different decades is about 4.3 to 3.6 days advance. Okay, but what my lab wanted to do is we wanted to use experiments to try to tease out which cues were driving this. There'd been some work using observational data, but we think that experiments are really best designed to untangle sort of these differing cues. We did some analyses that show if you use observational data, depending on how you're using it, which type you're using it, you get correlations between photoperiod and chilling, for example, of 0.3 to 0.9. So some of these correlations are quite high to be trying to run statistical models on with causal inference. But controlled experiments, obviously you put them in a box, you can decouple the cues, hopefully. Many of you have done these experiments, but for those of you who haven't, I'll just show you pictures of how they work so we're on the same page. And I'll do that with my former postdoc, Dan, who is here, and he's about to start one of these experiments. You usually start outside in the winter. This is in Quebec. You need to tag a nice little plant first, and he's about to take cuttings from this plant. So dormant woody twigs are what you need to get started. They have experienced some chilling for sure. So now they have some outdoor chilling. You bring them in and you put them in lovely flasks, and then they get put into growth chambers of different temperatures and day lengths, which gives you your forcing and photoperiod treatments, and then you wait for bud burst. So that is the general design of these experiments. And we wanted to do a meta-analysis. We wanted to get the answer that we would expect across all of the data, not just study by study with different treatments, for example. So we gathered data, all the data we could find in the literature on woody plant phenology, where we could calculate chilling and also calculate their forcing and photoperiod treatments. This is work done very much so with my lab over 2 long periods. This was a long project, so I want to acknowledge that they have done most of the work here and have been wonderful to work with. For the results going forward, we ended up needing daily data, obviously, to calculate chilling. So I'll be focusing on results from Europe and North America, which not surprisingly means most of the data we had. The experiments definitely differ in important ways. So we spent a lot of time trying to harmonize the data. If anyone ever wants to talk about that, I will just tell you briefly some of the different designs that happen. We had dormant samples. Usually they were twigs. Sometimes they were potted plants. Chilling could happen in controlled environments in this way, or generally outside, or sometimes a little bit outside and a little bit in a controlled environment. But either way, we calculated, I'll show you results using Utah models of chilling, but we have similar results using the dynamic model as well. And then forcing and photoperiod happens once they go into a warmer chamber. So chilling is a cooler treatment happening before a warm day length treatment as well, a chamber with light, and then you get bud burst. We've used Bayesian hierarchical models to try to estimate each cue robustly for every species. So this allows us to deal with the big issues we have in different amounts of sampling across species. I'll walk quickly through this model, but I'm happy to chat more about it. Basically, it's a very simple linear regression model at its base. We have day of bud burst has some intercept, plus the response to forcing, that's degrees Celsius, the response to photoperiod in hours per day, and the response to chilling, which is Utah units. We partially pool, and then we have an error model. We partially pool, excuse me, using phylogeny, such that we are better able to pool close relatives to one another using a sigma parameter, which effectively estimates the rate of evolution across our data, and also a lambda parameter, which estimates the importance of the phylogeny in scaling different species responses. I'm just going to take a moment to have a glass of water with the mic. Okay, so what did we find? we were really interested in teasing out which of the cues might drive declining sensitivities. So we were looking for whether there were strong chilling or photoperiod cues that we would see across species, or if some were just dominated by forcing, then they shouldn't have declining sensitivities. I'll show you the results one by one quickly. The phylogeny of the species that we have is here, and then I'm showing each effect size for the different species in change in days. The effect sizes are standardized, so you can compare them evenly across the different types of predictors. So you can compare photoperiod to chilling equally to forcing. So this is the forcing response. All of the responses in general were negative, meaning that plants tend to advance with more warmth. That's good. That's what we expected. You can see there's a fair bit of variability. Some species barely respond at all. Some advance 20 days. Oh, sorry, this is for chilling. So very big responses to chilling, and all species had responses. So we were excited. We were like, okay, all species have chilling responses. That would mean it could underlie declining sensitivities. Forcing had slightly, very similar, slightly smaller on average responses, but some species that had really strong chilling responses also had very strong forcing responses. And again, on average, everything was in advance in days with increased temperature. And then finally, photoperiod. There is an overall response to photoperiod, and it's definitely significant for those who do that type of thing. But it's quite small. And again, these are made on standardized predictors, so we can compare across these. What you see is that on average, it's much, much smaller than any of the other cues. So every species, for the most part, appeared to have a photoperiod cue, but it was very tiny. So it didn't seem like it could underlie declining sensitivities in any of the models that we ran. The other thing that's quite striking about it, and something I'm trying to parse out now as we work on this project, is they're very similar, as you can see. There is almost a uniform photoperiod cue for every species, except for a handful that are the oaks and fagus. And you might not be able to see it, but there is a dot and error right here. I should say these are means and 50% standard errors from the Bayesian model. This species is Fagus sylvatica. So even with a partially pooled model with other Fagus in the data, Fagus sylvatica is a massive outlier. So I would say there are questions we should ask ourselves when we assume that photoperiod cues from Fagus sylvatica may apply to other species, because even with our best modeling attempts, we're not seeing it even for other Fagus species. Other Fagus species certainly have strong photoperiod cues with or without this type of phylogenetic pooling, but certainly never as big as Fagus sylvatica. Okay, so we felt like we had nailed this. We had like a global analysis where we had looked at chilling, forcing, and photoperiod, and we could say that chilling is definitely driving, or could be driving, declining sensitivities, because we're seeing it across all of these species, and it's very large. We did one last thing, which we tried to forecast the data. We tried to forecast the model onto the European data where declining sensitivities had been observed, so specifically across Germany and Austria and the PEP75 data sets. First, we just looked at what the model would predict per degree of warming. So this is trying to adapt our linear regression model to a per degree of warming model. How much would you expect the days to bud burst to shift? And we have, if you do forcing only, again, remember it's that simple linear model, but once you add in chilling, you get this nonlinearity because of the way chilling accumulates. If you have both, you change that nonlinearity just a little bit. But when we forecasted it onto the sites with the PEP725 data, so trying to use their climate data to forecast bud burst, what we found was that chilling was not rapidly declining. So I'm happy to share, we have a larger figure of this in the paper, but effectively, if these dots were white, chilling would be declining a lot over time, but actually they stay blue. So in most of the sites we looked at, potentially chilling was increasing with warming because spaces, habitats were getting into these lower temperatures where chilling actually accumulates, because in many models, it may not accumulate below 0. And this meant that at 1 degree or even 4 degrees bud burst, we didn't expect to see this declining response. So we had done all this work, we thought we had nailed it, but then we actually, when we tried to project it onto the data, we figured out that as best we could tell, the current data is still in this effectively linear range. We had to go up to like 6 or 7 degrees Celsius of warming before we hit the nonlinearity for this model. And so that meant that my lab project, and this was a lab project where we all collaboratively worked on it, had spent 5 years and had all these ups and downs, but our main hypothesis for declining sensitivities in our minds had little support. So what's going on here? I want to go back to staring at the data, which is exactly what we did in my lab for several lab meetings after we looked at these data. And what we came up with was that we should have perhaps simulated a simple model to begin with. We do a lot of data simulation in our lab to fit Bayesian models, simply because we need to test our code. But this was more a question of, well, what does the forcing only model predict? So I simulated a simple model one day when I was on a train to go see one of my postdocs. I said, leaf out occurs as we understand it to occur after a certain thermal sum. So every day plants are accumulating warm temperatures if they occur, and at some summation of warm temperatures, they have enough heat or energy to leaf out. There was no chilling and no photoperiod in this model. And then I simulated it across a suite of different temperatures, and I'll show you those simulation results now. Oh, I should say this is a very simple model, if you're wondering, just to stress that. We have it formalized in a paper, but it literally is this many lines of code to simulate. And we didn't do it for 5 years. But these are what the simulations look like. Warming is again on the horizontal axis. I did little amounts of warming because I figured, Europe has warmed about 1.5, North America is somewhere in here depending on where you are. But this seemed like the range we'd be seeing in observational data. And then estimated sensitivity, again, in days per degree Celsius, this linear regression is on the vertical axis. Because we're doing advances in phenology, counterintuitively for some, declining sensitivities would be going up, just so you know. And what we saw was this. So with a model with only forcing temperatures, where we didn't change the forcing required over time or over warming, we had no chilling and we had no photoperiod, we saw exactly what we were seeing in our observational data. In fact, if you plot the observational data I showed you earlier on top, so this is silver birch from the PEP 725 data for those few sites, the 1950s and the 2000s fall perfectly in line. So this suggested to us that something perhaps simpler was going on, which is that we weren't thinking carefully enough about how we are fitting these linear regressions and how we're calculating temperature sensitivity. I will explain it to you the way my colleagues finally did that was most simple. I have lots of other ways to explain it, such as the amount of time or the way plants count time is changing. Okay, this is a great moment for my cartoon of grandma. So the way you can think about this and the way I think it's easiest to think about this is an analogy with grandma who lives 200 North American miles away and how fast you get there, the hours it takes you to arrive at grandma's house from when you leave versus how fast you go. So we all know that if you go very slowly to grandma's house, it would take you much longer to get there than if you drove very quickly to grandma's house. And we are probably not necessarily that surprised that this is a non-linear relationship where increasing your speed, for example, by 5 miles per hour, if you're going 10 miles per hour, means you get there twice as fast. But increasing your speed 5 miles per hour at 50 miles per hour means you only get there 10% faster. So how quickly you're changing depends on the speed of the process. And this, I would argue, is exactly what we're doing to plants and many other organisms with climate change. So spring temperatures are accelerating because warming is acting as an accelerator. So we're speeding up processes. And as it gets warmer and warmer, we should expect what look like slower and slower rates for any process based on a summation such as this, which is a lot of different biological processes, I would argue. So effectively, what's happening is linear approximation, which does work fine for nonlinearities, but only within a range, starts to break down. And you'll see that here I'm showing you a much bigger range of temperatures than I've shown before. It's much easier to see this problem across it. And so what works here is a very steep slope at cool temperatures where 1 degree of warming makes a big difference if you calculate it in change in days per degree Celsius, is obviously not what you'd see at the other extreme of these data, where 1 degree of warming makes a very small difference at 30 degrees. And so you get higher estimates at cool temperatures and you'll get lower estimates at warm temperatures. And I see this often across spatial data sets and temporal data sets, where I think we need to train ourselves to question the idea of smaller responses in warmer locations if the underlying process is some sort of threshold model. Okay, so how do we fix this other than just thinking about it? Well, there's a very simple fix for anyone like me who does a lot of linear regression, which is that the natural change for this is the natural correction for an inverse is the log transformation. And so these are the simulated data where we've log transformed both the X and the Y axis, and you can see it linearizes the relationship. We've also tested it on a number of long-term data sets. In fact, for every long-term phenological data set that we've looked at, we find evidence of declining sensitivities. And we also do not find that evidence once we use a log transformation on the data. So it doesn't seem to be a prevalent trend if you log transform your data. I think the better option is the much harder option, which is to build a generative model that includes climate change itself and try to understand sort of the simplest generative model we could have that includes climate change. This is a project that is almost done. It's led by my colleague, Jonathan Auerbach, and also worked with Andrew Gelman to try to build this. And we're hoping to have it done soon, but it is quite a larger ask to try to get these models to run based on the underlying data. I think they bring up a different answer, which is how we do science, at least the ways that I was trained, really makes it easy now with climate change to pick up some of these artifacts and not clearly have a process to know when they may be statistical artifacts versus they may be biological reality. Our results don't say whether plants are shifting their responses to warming. They only say that one reasonable explanation is that the underlying statistical model wasn't a good fit for the process that we were using. But I do think that we have non-identifiability in a lot of our models that we use and we don't recognize it. You can have it both in your model and you can have it in the combination of your model and your data. So a lot of those correlations I talked about between photoperiod and chilling, for example, might mean that you have a model that seems functional, but once you put in correlated data, it's non-identifiable at the end of the day. And I think that's a big problem where we need more data. That's something we're trying to do in the lab by getting more microsensors and also just fundamentally better data, bigger experiments, et cetera. I also think we should be simulating more data and racing the same data. So we should all have standardized data sets that we use on our models so that we can cross-compare our models more reasonably, so that I can cross-compare my sort of simple Bayesian linear regression with more advanced models that have many more parameters. And I think that's something we should try to get the field to do. We're starting that in a small part with my collaborators, Jonathan Auerbach and David Keplinger from George Mason University, these tiny little GitHub pictures here, by asking people to help us predict cherry blossom blooms at 4 locations around the globe. It just finished. You're supposed to submit your application by the 28th of February, but it's getting to the point where Washington, DC blooms are so early, we might need to move that earlier. Okay, and so now I have told you how I think there's good evidence that climate change is accelerating biological time. And what I think is careful that we have to remember here is that we're effectively accelerating biological time when we continue to use calendar time. So the unit of day, I would argue, is fundamentally changing for animals, plants, and all organisms as they warm up. They certainly are using that day length, but if temperature is the major driver, we have to remember that that unit of day is not constant. It's non-stationary itself for organisms. And I think that has a really big impact on how we think about climate change models themselves. And we should be careful to use them. But what I want to do quickly is just tell you, since I am here visiting, I would love to chat with people. I generally love to chat about what I'm working on next, although I'm happy to talk about any of these previous projects. Some of them are still getting written up. Really comes back to this slide. Many of the things I talked about, we're still doing lots of work in the lab on trophic mismatch. We have a big new global analysis of that coming out, which basically suggests that there's no widespread evidence of trophic asynchrony, but there is a lot of trophic differences across clades. And we're really interested in trying to understand whether extended growing seasons actually impact how plants grow and thus would cascade to impacts on carbon storage. And then finally, one of the things I'm hoping to do with Isabel as well, I do work on wine grapes a lot, which I didn't talk about today, but to better understand the difference between as seasons lengthen and plants can grow more, the sort of mean shift that we think about, versus these discrete events where you can have massive losses of tissue, but depending on the exact phenological stage that they hit at is one thing I'm really interested in. And we're trying to scale up and use these cues that we now better understand to try to ask, okay, if we have general models that suggest that tracking could matter to how communities assemble, and we understand the phenological cues, how do we tie these pieces together? And for us, that's been trying to work at the sort of in between proximate and ultimate level and say, what are the drivers of the cues themselves? And so for communities like forests that I work on, there's this really big discrepancy in what plants are experiencing from the early to late spring. So this time axis is just early to late spring. I have some example species. This is a birch, an oak, and a fagus to sort of temporally time you. And the big difference, I would argue, for species early is that they have these massive frost risks. So they are often getting hit by frost. They're losing tissue to frost if they're early. So why ever be early? Because late seems better. But early is a great opportunity where soil microbial communities are turning over. That means lots of soil nutrients are available. Obviously, it's a highlight environment in deciduous forests. And so you get a really early access to resources if you're early. It seems like it could be a good idea to be early. So you could be risky and early, or you have to be competitively dominant and late, I would argue, if you want to be conservative. What we're doing now in the lab is collapsing both the BN and the TRI database to use them as trait databases to try to also combine them with our meta-analysis of phenological cues and look at whether you see predictable trends in traits that would follow this pattern of species are being risky and early, have maybe cheap tissue traits and are shorter, for example, versus later active species should have different stem densities, different leaf traits that maybe it's harder to lose the leaf, but they help the plants draw down resources better. They should certainly be taller so they can get access to lower resources lower in the soil. We have a joint model where we fit all of this at once. So we fit the entire TRI and BN databases at the same time that we fit our lab Osprey database. And what we've looked at is several traits for which we have enough data. I'll just show you one here and one cue. So height of a bunch of different tree species, again, where we had enough data, is on the horizontal axis. And the chilling cue from this model is on the vertical axis. As you go down, it gets stronger. And we generally think that chilling cues would make you have later leaf out. So you can think of this as an axis where early species are at the top, later species within the spring are at the bottom. And what we do see then, obviously, is this trade-off that we might predict, where early species are these sort of shorter species, and Fagus and larger, taller trees have these stronger chilling cues that allow them to leaf out later. So we are seeing correlations that suggest this risky and early versus late and conservative is happening. What's interesting to me, though, is that means that if the traits in these forest communities are structured such that perhaps frost risk is one of the big things that species differ in how they respond to, we're fundamentally changing it with climate change. What is risky and how risky you are early in the season now is different than it would have been 50 or 80 years ago as a species. And we've shown that predominantly by my student Kat Chamberlain, that in Europe, we have strong evidence, this has been done in other labs as well, that frost risk has been increasing in the last 40 years, and that where you get frosted, how close you are to the coast, how much elevation affects it, has also shifted. And we've shown that that can cascade on to impacting plant growth, and that some plants really do have this trade-off. So we're fundamentally shifting the trade-off landscape, which I think is really interesting. And that couples with what we want to better understand in an experiment we're starting this summer, which is that do these longer growing seasons actually lead trees to grow more? And by trees, I mean saplings for this experiment. So we're going to have both longer growing season treatments and also event treatments or discrete events. So we're going to have both drought and defoliation treatments that will occur at specific phenological times. So we can try to both estimate when are events worst for different tree species in terms of impacting growth. And also what's the relative impact of an event like a major drought during bud burst or mid-season versus a longer growing season? How do we compare those sorts of impacts? We're doing this by trying to also have new microsensors, microdendrometers with a lab group in Oregon. And we're hoping that eventually we'll be able to scale this up to reanalyze tree ring data and try to look for patterns of shifting tree ring growth with phenology. That is the big snapshot of just some of the projects we're working on next. I want to thank my amazing lab, some of whom are shown here, as well as wonderful collaborators who have been critical to these projects. And hopefully we have some time for questions. Thanks.
Thank you very much, Lizzie. Are there some questions in the room? First, on Zoom, not yet.
Hi, thanks for this amazing, very insightful. I was wondering, it seemed to me that there was a correlation between your chilling, forcing, and also to some extent photoperiod cues between species, I mean across species. And maybe I missed something, but can you explain it? Can you explain why you get this correlation?
So there isn't actually a strong correlation across all species in their chilling, forcing, and photoperiod cues, but there are in certain clades. And why you get them, I think, is if you want some of it, and who knows, I think this is a great question. But if you want to be ultra late, there are certainly species that appear to have just a strong photoperiod cue and not a strong chilling cue to be ultra late. And then there are species that definitely have both. They have a strong photoperiod and a strong chilling. But we have looked at the core. So we've looked at these correlations in different data sets. There's certainly a set of species which is often predominated by European and North American, sort of the classic temperate deciduous species, where that is happening, where you see correlated responses as opposed to a trade-off between the cues. But in general, the earliest species have weak photoperiod, weak chilling, and strong forcing. And that sort of pattern shifts across the different species to get to later leaf out.
Any other questions? May I ask one? So that's great that I see that you are now looking at the relationship between other traits, functional traits, with the responses to chilling, forcing, etc. And coming back to what you did during your PhD in basic species, where you show that those species tend to shift more their phenology. Do you have an idea now of why? I mean, if there are syndromes, trait syndromes that makes invasive more responsive to climate change?
That's a great question.
Because of all
when we got into the trait question, because the invader research became so fixated on phenology as like a singular trait, that phenology would explain why invaders are so successful. But certainly it's super correlated with a bunch of other traits, was my sense from just like the certain species that I have worked with. So I have not worked enough continually with invaders since then, and I haven't seen enough compelling evidence to say that there is a syndrome for invasive species that makes them invaders, of which one part is phenology. But I suspect that there are these other traits like fast growth, shorter roots, things that basically make you cheap and fast species, seem to correlate with early phenology. So I don't think it's necessarily just phenology that makes species successful in new communities. That said, my student Dan does have work where in a greenhouse, we looked at the impact of being early on the competitive outcomes. We shifted the chilling cues and said, if you can get an invasive and a native to be at the same time, how different is the competitive outcome than if you let the exotic species be earlier by some natural amount? Because basically they have no chilling cues, which in germination we would call stratification. And in that case, we saw that the majority of the competitive effect is due to phenology. So there's certainly something going on where these priority effects from phenology matter, but I doubt that they're the only piece. In the work we're doing with trees, they're all native species, but I think we are seeing some patterns. That said, the patterns I showed here are from a meta-analysis. We don't have that many species underlying them. And we have a separate analysis where we collected all of our own data across 8 different forests on both the east and west coast of North America. And we have 40 species, and the patterns aren't as striking. So I think the answer is probably super complicated. I always feel like I gave a good talk if people ask questions, not to pressure you or anything. Thank you.
So thanks a lot for your talk. It was really great. I'm not a plants ecologist at all, but I'm more working on animals, and I really liked the last figure you showed. There's a correlation with the height of the trees and the sensitivity like to frost. And also I was wondering, for like more the population persistence, actually it's maybe not a problem to being small, fast-growing species, and then okay, maybe you have this risky thing with the frost, but it would be a problem. It would be more like a long-living species that are having this. So maybe it's. I wonder if you're going to make models of more population dynamic to understand the consequences of this kind of relationships.
I, as a community ecologist, am probably not going to do population models personally, but I do think that there is some of a correlation probably with lifespan, that the species that are the earliest never have the longest lifespan. So there's definitely some trade-offs there that are happening in terms of being fast and early. And I do think these exact processes have been shown in the great tit and other animal populations where the individuals that arrive early are taking higher risks. The individuals that arrive later are sort of losing out, and there is a sweet spot in the middle that all the species are trying to work towards, and there's usually high selection on that sweet spot. I think there's probably far more interesting dynamics going on about, if you look over the long term, when to be early versus late within a population, because then you introduce really interesting dynamics of bed hedging. And I think we're in a bad spot with bed hedging in ecology because we just don't have long enough data to really understand and predict bed hedging, is my personal sense. Like for the annual communities even, they are usually existing in super highly variable areas of environment where I would argue we need like 100 years of data, and we have at best 25 or 30, at least in the US. So I think really interesting bed hedging starts to happen. I think it's hard to know how to merge it with observational ground data.
So we have a question online by Christine Ménard.
Hi. Yeah. So you started by saying that most of the data sets that you were doing the analysis come from North America and Europe. And you showed that sensitivity to the temperature increases is lower at higher temperatures. And so I'm wondering if you expect any difference with tropical species, and what would that mean for tropical communities with climate change?
Yeah, that's a great question. Just to clarify, the sensitivity as defined as days per degree Celsius appears lower at lower temperatures, but I would argue there's no actual change in how the plant is responding to temperature. It's a totally predictable response, and the underlying biology hasn't shifted. So I don't think that gives us any insight, that sort of relationship gives us any insight into what should happen with tropical species. I do think for tropical species, we get much more into there is an actual nonlinearity in temperature responses, where high temperatures usually lead to much slower responses, but up to some point, responses speed up. And so I think in the tropics, you get into some of those areas where small differences in temperature can actually have much larger responses, but you eventually might get to some sort of tipping point where the responses are too hot. That said, we do not have a lot of data on the tropics, and I have a new global analysis of trophic asynchrony. And in that analysis, we don't see a strong latitudinal effect, but we also see what I think we don't talk about enough, which gets back to your question about population variability, which is that these populations have high interannual variability, at least in the temperate zone. And we don't have good measures of that. We don't have enough data usually to accurately estimate variance over time. And we have so little of those data in the tropics that I think we make a lot of predictions about how small temperature degree changes should have the impacts. And they should, from all of our fundamental physiological data. And we see some of those trends, but we just have a lot more data in the tropics before we could start to make predictions. And I would say specifically because we have very poor estimates of response to temperature and natural variability that these populations can be buffered with.
I'm just looking at all these Erlenmeyers, and I'm trying to figure out how one does the experiments looking at the chilling effects, the temperature effects, and the photoperiod effects. Is that typically a fully crossed experiment? So you have a range of chilling temperatures, and for each of the chilling temperatures, you look at different temperature ranges, and for all the different chilling and temperature ranges, you look at the wavelength effects, or how does that work?
Yeah, lots of great questions embedded in that. A simple answer I will say is that we did look at this, and we published it last year or a year ago in New Phytologist. We just looked at actually like what are the experimental designs that are out there. we didn't actually look at like the three-way cross. Well, actually we did. Three-way crosses where you actually have a full cross between all your forcing treatments, all your chilling treatments, and all your day length treatments are extremely rare. There's like 4 out there, maybe 5 out of about 80 different studies. So the beautiful picture is because we did a fully crossed one. So we had many, many flasks and we had over 2,000 cuttings. So they're really hard to do as fully crossed. And in this experiment, we had only 2 levels of forcing, 2 levels of photoperiod, and 3 levels. So they're never approaching like a response surface design. So there's some big problems there. I think we don't have, in this paper, those may not have noticed, but my lab and I struggled deeply with trying to estimate the interactions between chilling and photoperiod or forcing and chilling. We didn't estimate them because we felt the underlying data wasn't robust to estimate interactions. It takes a lot, usually takes 16 times more replicates to estimate an interaction than to estimate a main effect. So I would say the main effects that you see are smearing over those interaction terms. We know for chilling and forcing, they're probably big and we're missing them with the data we have. And then we have lots of cool studies on Arabidopsis that, day length may matter, the sun, how the sun has certain things at sunrise might matter. That said, these are by far the biggest cues for woody species. So I feel confident that we're getting the largest part of the story and we're not missing like a critical radiation type of cue. However, the one big thing I think we are missing is that the model of chilling, I spent a long time telling you that the chilling treatments are treatments that come before forcing. And that's because that is how they are defined. And there's an underlying physiology that something has changed from the plant from eco to endodormant. But we, I would argue, effectively have no good idea of what chilling is other than something we name a treatment. And so all of our estimates of chilling and forcing, I think, are based on poor estimates of what this winter cool is, that obviously is some sort of metric that plants are using, and they're using it as a sort of way to stop responding to winter cold and eventually respond to spring warming. So until we understand what chilling is, which is a process that happens very slowly at very low temperatures, so it's not a lot of fun to do molecular biology on, I don't think we have good estimates of chilling and forcing. So I think the estimates I'm showing you here are chilling and forcing in air quotes, but they are not robust estimates of what the plants are probably doing.
I think we all set. Thank you very much.
Thank you.
Lizzie.